Hi everyone, today I would like to talk about how we build a multi-scale metabolic model to assess the metabolic impact of water limitation in sorghum. Sorghum is a C4 plant that can produce very high biomass. And normally we grow sorghum in marginal soil, which has very limited water and nutrition. And in this project, we are trying to we are trying to improve the biomass production in the limited water conditions. But there are scientific challenges to improve biomass yield in drought, because we don't understand very well about the plant metabolism. Plant metabolism is very complicated network, and it's a highly regulated metabolic networks. And plants live in a dynamic environment and are affected by multiple environmental factors such as light, water, and temperature maybe at the same time. And to address those challenges, we are trying to develop a genome-scale metabolic model to simulate the plant metabolism. This kind of genome-scale model I will introduce later. And we are trying to integrate a C4 biochemical models with the genome-scale metabolic model. And the objective of this model is trying to simulate the impact of drought metabolism. And the second, we are trying to identify key reactions that limit biomass production under drought stress. And first, we are trying to reconstruct the genome-scale metabolic model of sorghum. We use the database from MetaSeq, PlantSeq, which is and is also called a plant metabolic network, and pathway tools. Those database they have the individuals metabolic reactions using automatic computational pipeline, homemade computation pipeline, which is called Ravi, to construct the genome-scale metabolic model of sorghum, and then we collect other data such as the transcriptional data, metabolomic data, and the biomass composition, and also the carbon dioxide and the light, trying to constrain this genome-scale metabolic model, and then we do the flux balance analysis and other and to see what is the carbon distribution and metabolic flux distribution in this complicated metabolic network. And to better simulate the drought impact on the plant metabolism, we combining a C4 biochemical models with this genome-scale metabolic model. And this C4 biochemical model is the input of the physiological data, and then the output is the reaction rate of those key reaction of the photosynthesis, and we use this output of C4 biochemical kinetic models as the input of the genome-scale metabolic model to constrain the key reaction of the photosynthetic data, and the output is the biomass. So and the reaction flux of this metabolic network. And then here is our experiment design to how we how we perform the drought stress. We grow those small sorghum for 14 days, and then we stop watering them, and we for 7 days. After 7 days, we start watering them to once a day to 40% soil water content. At the same during the drought treatment, we collect the plant samples for insect data and metabolomic data analysis, and then we also measure for physiologic data and biomass and biomass. And we found that in the first 6 days after of drought treatment, we don't see a significant difference in the net photosynthesis rate between the well water and the drought conditions. After 7 days, when the soil water content dropped below 30%, the photosynthesis rate in the drought group dropped significantly, and the biomass production did not change until the day 10. And we collect the transcriptomic data, transcriptomic data, and the metabolomic data. And here is the PCA analysis for the IAC data, and it clearly shows that after day 6, they already show the separation between the well water group and the drought group. That means the plant start to begin to respond to plant even in the transcriptional level and the metabolic level even before the photosynthesis rate decrease. And then we are trying to use the modeling to show what is going on in the plant metabolism. If you look at the figures in the middle, our model predict that the biomass production rate begin to show significant difference in 35% water compared to the well water group. Even though in the 35% water condition, the photosynthesis rate do not show the significant difference between the well water group. And that is validated and confirmed by our later experiment using 3 conditions: well water and 35% water and 25% water. And our model is trying to find out why what's going on in this like a moderate drought stress, and what could be the limiting factor. And we found that the energy could be the limiting factors. In our metabolic model, we found that the total ATP production in the photosynthetic process drop significantly in 35% water, but at the same time, the total energy cost to fix per 1 carbon from carbon dioxide increase under drought condition, and then leading to the result that compared to the well water condition, that in the drought the plant has a less energy supply for the biomass production. So we come to a hypothesis that if we increase the light intensity, we could improve the biomass production under 35% water condition. And then we design an experiment, a highlight experiments that are trying to improve the biomass production. So we give the plant during the drought treatment, we give them highlight treatment. So in the low light group, we found that after 14 days after 14 days of drought treatment, they show significant difference between the well water group and the 35% water group. But in the high light condition, the 35% water group actually the biomass concentration increase, and they have a similar level to the well water group. Here we come to a summary. We build a multi-scale metabolic model framework of sorghum and combining C4 biochemical models and the genome-scale metabolic model. And this multi-scale metabolic model predict that the energy is actually the limiting factor for biomass production in moderate drought condition in sorghum. And the modeling prediction was confirmed by experiment. And that's and our hypothesis is confirmed that high light can alleviate the biomass reduction under moderate drought condition. Finally, I want to thanks to my previous advisor Suri and our collaborators. And here I want to have a advertisement that we are hiring a postdoc right now in my group. Our group is newly built in the Shenzhen Agricultural Genomic Institute, Chinese Academy of Agricultural Science. Whoever interested in plant metabolism and whoever interested in computational biology are welcome to send reach to me to my email zhaozheng01@caas.cn. Thank you.